topic is different and nothing much to write. There is some problem with these things. The topic is differential leukocyte count, as you will see in your syllabus. Differential means what is the meaning of the term differential? Nothing much to write. In most cases I will say nothing much to write. Just follow a standard physiology practical textbook. Okay, there are books, several textbooks in the market by Dr. Debashish Pramanik, by Dr. A.K. Jain, by Dr. C.L. Ghai. In my opinion, that is only in my opinion, there is no hard and fast rule. Dr. A.K. Jain should be good for the beginners because it's a colorful book, salient points are written. In my opinion, this opinion will vary among faculties and will vary among your. This is a professional course. There is no hard and fast rule. The objective at the end of the day is, at the end of the year is, you should know the topics, you should have the knowledge and you should perform in the exam. There is no hard and fast rule. But this is my personal opinion after going through some books, that it is for the beginners. The book by Dr. A.K. Jain is very suitable, but there are so many books, you can follow anyone, you have to know the topic. And I also recommend the book by Dr. C.L. Ghai, because there are so many questions and answers are given at the end of each topic. So after you perform the topic, the examiner will go you and ask how you have done, and there are related theoretical questions will be asked. So in the book by Dr. C.L. Ghai, there are many such questions and answers are given. After you know about the topic, if you read those, then that should help you. That is my recommendation. But there is no hard and fast rule that I have to follow this book or something. You have to know about the topic, that is the aim. So whatever books you are following, you have to learn the topic. Okay. So the topic given in the what we told, that whole syllabus, every subject is divided into competencies. So hematology is, in physiology it is competency serial number 2, PY2. Now in PY2 dot something, say 2.10 or 2.9, I think that topics which you have to cover in the practical, the topics you have to do hands-on in practical, those are given. So one of those topics is differential leukocyte count. So what is the meaning of differential? What is the meaning of the term differential? You don't have to stand, you sit and say. Distinguish, yes. Differential refers to something that based on some difference. So leukocytes is, as you have some, I think have some preliminary knowledge, leukocytes, the white blood cells in the blood are termed as leukocytes. Okay. Now differential leukocyte count is, there are different types of leukocytes and we have to distinguish them and count them. Count them means it is usually expressed in percentage. That out of 100, how many is this type, how many is this type, this we have to count, and there are normally accepted counts, and we have to find whether that blood sample, that is from a particular person, particular subject or particular patient. What is the difference between subject and patient? We just told, diseased, someone apparently diseased or who is coming to us for some symptoms. So whenever we learn any topic, there will be some associated words or associated terms. So come symptoms. What is symptom? This will be again dealt in the clinical physiology classes and clinical. What is symptom? Symptom means the person who is coming for some medical advice or some medical consultation from the medical professional. He or she speaks of some, I have this, I have fever. He or she will come and say, I have fever, I have some headache. So whatever he or she complains of, that is called the symptom, and whatever the clinician, the medical professional elicits is called a sign. Like one classic example is, the person will come and say, I have some pain in my belly. Now what the clinician is supposed to do? He or she is supposed to examine the patient subject. Before identifying a disease, it is better to call the person, that individual, a subject, not the patient. So examine the patient, or examine the subject is made to lie down. Now the clinician puts his hand over the belly and examines the patient. Are you feeling the pain here? Okay, the person says yes, or the person makes some sound after feeling that pain. Now this, which the examiner, or the examiner means the clinician, the medical profession elicits, is termed as the sign. So pain is the symptom, but this sign of eliciting that pain complained by the subject is called tenderness. The term is tenderness. So pain is the symptom, tenderness is the sign. Okay. So these are some initial terms you have to know. So difference between sign and symptom, difference between subject and patient. So in the blood sample, from the blood sample of a particular subject, whatever is there, we are counting the different types of WBCs, and what types of, it is expressed in percentage, and how many WBCs out of 100, this type of WBC is say 50, another type is say 20 or 22, another is say 5, 7. So what are the types of white blood cells? We have these will be discussed. Whatever is in your course will be discussed in the theoretical classes. Already hematology theoretical classes have started. On the first day you had a class about, on Monday, this Monday you had a class on blood components and plasma proteins, I think. That was the topic, plasma proteins. So next topic will be on, you have to learn about hemoglobin. So next there are topics, how RBCs, WBCs, these white blood cells. So as we know, blood components are red blood cells, white blood cells, platelets. These are the cells, and blood is composed of plasma and fluid. The liquid portion is called plasma. What is serum? Plasma short of clotting factors, coagulation factors, also called coagulation factors. So that is serum. This is from your initial preliminary school level biology knowledge. Okay. So plasma minus clotting factors is serum. So, and blood has plasma almost more or less 55%, more or less, and 45% is the cellular components. What are the cellular components of blood? Red blood cells, white blood cells, and platelets proper, if there are, except abnormal cells. If in a disease subject there are abnormal cells, we have to speak of that, but normally, physiologically, physiology is the study of, it is the study of the normal functional part of the human body, normal functions. Anatomy is about the structure, everything is related. These divisions are for the convenience of gaining knowledge. Everything is related. Anatomy is about the structure, physiology is about the functional part. So normally, physiologically, you will use the term physiologically, and the corresponding term will become a term pathological. Pathological means whatever is happening we are noting in the disease state. That means abnormal sense is blood, that is cells in blood, that is pathological. If you don't have interest in the class, then you may leave. Don't disturb the class. From the initial moments I am watching, you have to talk about yourself, you leave the class. No need to attend the class. Maintain discipline. So, blood components, plasma 55%, cellular components physiologically, that is normally, not in the disease state, 45% these cells, red blood cells, white blood cells, and platelets. White blood cells are of 5 types, but again before that it is classified as granulocytes and agranulocytes. Three of these types will contain granules, which are termed granulocytes. 2 of them will be divide of granules, which will be agranulocytes. Granulocytes, now what are the names? Granulocytes end with a suffix. Suffix means at the end of the word, whatever it ends with the suffix, it has the suffix fil in each case, neutrophil, eosinophil, basophil. Okay, agranulocytes are lymphocyte and monocyte. Okay, lymphocyte and monocytes. Okay, lymphocytes are further classified according to size on small lymphocytes and large lymphocytes. Now all these types you have to count in you count 100 cells, or if it is possible to count 200 cells, then how many are neutrophils, how many are eosinophils, how many are basophils, how many are lymphocytes. Usually in the standard laboratory reports we have lymphocytes, we don't classify again as small and large, but there are lymphocytes can be distinguished, some are smaller, some are larger, so that can be distinguished. But in the accepted standard, in the biochemical reports, investigations, neutrophil, lymphocyte, basophil, eosinophil and monocytes. Okay, out of these, lymphocytes and monocytes contain no granules, and neutrophil, basophil and eosinophil contains granules. Now obviously we can't see this under the naked eye, we have to examine these, all this is to be done under the microscope. Manual methods under the microscope. Nowadays there are automated methods, but we should, for beginners, for the basic ideas, we learn about the manual methods. So for that, blood sample has to be stained first with some material, so that the cells can be visible. After that, cells have to be identified and then counted. So the topic is differential leukocyte count, but for that, first part is, what we have to know about is how the blood sample is collected. Now where is blood contained in the human body? In the blood vessels. Blood vessels are of three types: arteries, veins and capillaries. Arteries are relatively deep seated and veins are superficial, capillaries are rather more superficial. Capillaries are junctional parts of arteries and veins, at the junction of arteries and veins. And capillary blood, all these will be done in the theoretical part also, but capillary blood, what you have to know, capillary blood is more or less more towards arterial blood composition. Venous blood contains more deoxygenated, then venous blood is more deoxygenated. At the capillary level, the tissue, the cells are receiving oxygen, carbon dioxide is going out from that, oxygen part is very less in venous blood. So arterial blood, capillary blood has more or less composition that of arterial blood, and hemoglobin cells are somewhat more in amount, and in the venous blood it is somewhat, venous blood is more accurate. In accepted standards, venous blood, the counts in venous blood are more accurate. But there are few hematological experiments which have to be done with the capillary blood. It is not possible with the venous blood. Some are possible with both capillary blood or venous blood, but there will be few experiments, which has to be done with capillary blood taken from the capillary. Now how to collect the blood? First thing is how to collect the blood. So to collect the blood you need a needle. Okay. So these things you can see, everybody can see. Just you will work with your own hands, but these are called lancet needles, named as lancet needles. These are our classical. The needle is termed on the bore of the needle, 23 to 25 bore needles. These are classical needles. These needles are somewhat the pointed end is somewhat more thicker. Okay, so anything can be used. Mostly we have the needles supplied here at these lancet needles. You will work with those, but I recommend you can bring your own needles also. The precautionary part is, one needle is exclusively for oneself. This is very common sense that one needle you cannot use on different subjects. One needle for oneself and to be discarded after a single use. Okay. That is the rule. Okay. For safety, for your own safety. So if you can carry your own needles, it is better you use them and discard it. Here you will be supplied, most needles are supplied, which are these are lancet needles. So how to collect the blood? You have to make a deep prick, 2 to 3 millimeter deep on your own finger. Usually the middle or ring finger is used. So where capillary blood is more, it is more possibility for the capillary blood to collect. That is over the fingers, near the tips of the fingers, on the ear lobes, in cases of children, from the heel region. Okay, these are the common areas from where capillary blood is collected. For your purposes, your experimental purposes, you have to use the needle, make a deep prick. In the beginning you will have some apprehensions that it will be painful. It is very little painful, but you have to make a deep prick. If you don't make a deep prick, what will happen? What is there outside the cells, between two cells? What is there? Cells comprise tissues, there is tissue fluid. So if you don't make a deep prick, that is, maybe some blood will come, there is some rupture of the capillary, some blood will come, but it will be mixed with tissue fluid. So then you can't see the cells. Whatever is required, our purpose will not be achieved. Cells will not be seen and you can't count and you can't get your experimental result. So prick will be one single prick and deep prick. Now blood will come and it can be identified just by looking that blood is coming out, that is not mixed with tissue fluid. So one drop of blood is to be taken over a glass slide. So how it is taken? The glass slide is placed on a horizontal surface, blood is coming out directly from the finger. You can put the blood over the glass slide, where it is to be kept near one end. Which end? The shorter. This glass slide is rectangular in shape, over one shorter end. That means the breadth of the slide, not along the length of the slide. In a rectangle, the shorter arm is called the breadth. Near one breadth, you have to put the drop of blood. So this slide is kept on a horizontal surface, and another slide is to be used. This slide is termed as the spreader slide. That means that blood drop will have to be spread over the slide on which the blood drop is kept. So suppose the blood drop is at the one end, so you have to put this slide over that blood drop. Again, there is the procedure to be followed. If you put the spreader, this is now termed as the spreader slide. If the spreader slide is put directly over the blood drop directly, then there is some possibility that it may spill. So what we do, we put it a say 1 millimeter in front of this and holding at an approximate, more or less at 45 degrees in the front of that drop, and then over this slide we just we come back, we put it at the front, we come back and it will touch the blood drop, and now the blood drop will get uniformly spilled. We are not to put it directly over the blood drop, there is chance of some spilling. So we put it in front of the blood drop, draw the slide backwards, spreader slide backwards. Now it touches the blood drop and it gets spread over this edge. This edge which is on that slide, this edge of the spreader slide it gets, and now we push it. One single push. This slide is on a horizontal surface. One single push. We should not hesitate. One single push and it will get more or less uniformly spread over this first slide. The second slide is called the spreader slide and it will get pushed on the first slide, and we will get whatever we get, that is called a blood smear. After it has been spread, it is called the blood smear, prepared from one, or maybe if the drop is small, then it is maybe 2 drops, then it is sprayed on this one glass slide. This is called the blood smear. Whatever you have to see, you have to whatever experiment you have to do with the help of this blood smear. So first part is collection of blood sample. Now before that, before the collection of the blood sample, before pricking, what are the steps to be followed? This is how to collect the smear about the blood drop. What are the steps to be followed? So from common sense, the part of the body from where you are taking the blood should be clean. So what should be used for that? Usually we use this spirit or alcohol, like at least since COVID days we know how to sanitize the hands. Okay, so that is just cleaning purpose. You have to use spirit and cotton. With the help of spirit and cotton, you just rub that area of the body, whatever. So when you have to collect blood from children, what will say? Usually the heel of the very small, say infants, just newborn babies, heel is used. For yourself, on adults, you prick the tip of the finger, usually the middle or the ring finger. So you rub it with cotton using spirit and allow it to dry. Now this allow it to dry, you were asked in the exam as well as for knowledge, why we are allowing it to dry. Spirit, after rubbing with spirit cotton, like after sanitizing the hands, we allow it to dry before touching the food material. Why? Because this disinfecting of that part of the skin takes some time. So we allow some time. That time, by the time it will get dried up, the part has become disinfected. My purpose, cleaning the hands, that is the reason why we are allowing it to dry. One reason. Another reason is, if we don't allow it to dry and prick, then blood will get mixed with that spirit. Before drying, blood will get, and blood should not be diluted. If the blood is diluted, counts will be erroneous. That means it will be falsely low, right? Do you get it? If blood is diluted with something else, then the counts will be erroneous. So blood should not be diluted, either with tissue fluid, so the peak should be deep, so that it is only blood coming out and not getting mixed with tissue fluid, and it should be done after that spirit has dried. The purpose is to disinfect, primary purpose. But why you are allowing it to dry? For allowing that time for the disinfection to occur, and also so that the blood does not get mixed with this spirit, okay, or alcohol, whatever we are using. Okay, so after cleaning the finger and pricking, collecting the blood drop on the slide, preparing the smear, now that smear we can't identify just by preparing the smear and seeing it under the microscope. It can be seen, but cells cannot be identified. For that, staining of that smear is required. So first is collection of blood sample, then preparing the smear, collection of blood sample with necessary precautions, disinfecting the fingertip. In your case, it will be the fingertip when you start with these experiments. Fingertip, disinfecting that, collection of the blood sample, preparing the smear by collecting the blood drop, collection of the blood sample, then preparing the smear, and then staining the smear. For staining the smear, for identifying the cells and staining the smear, what we use is called, the stain is called the Leishman stain. Leishman stain looks like this. It is deep blue in color. Okay, this is called the Leishman stain. Leishman stain, after the smear is collected, Leishman stain is to be put over the smear. Just before that, how does a uniform smear, ideal smear should look like? It should be tongue shaped. Like we are drawing it from one end, where the blood drop is put and sprayed over the edge of the spreader slide. We are drawing it then forwards. So as we go towards the other end of the slide, the smear, the blood will, this film will taper. That means it will look like a tongue shape. And ideally, practically it will not happen, but ideally it should be almost 2/3 of the slide, almost, more or less 2/3. It is near one end and more or less towards 2/3. Okay, more than half, almost 2/3 of this first slide, it will be spread and it will be tongue shaped. It will not be as parallel to the edges of the length of the slide, it will be tongue shaped. As we are going further, the edges are blank. So the blood will be, the smear will be tongue shaped. Any book you follow, these pictures are given. Any practical textbook you follow, these pictures are given. It will be tongue shaped. Now we have to put Leishman stain over the smear. Now this is just drop by drop we put using this. Drop by drop we put Leishman stain over the smear. Now how many drops? There is no hard and fast rule. How many drops? We will put it drop by drop, so as to we will look at it enough to cover the smear. So at first it will not be that in the first smear I make it exactly uniform. It is not human, this physiology or human body or anything it is not like mathematics 2 and 2 plus 2 4. So first day your smear will not be that good. It is not expected that maybe it is half of the slide, so it requires some 1 or 2 drops less. So as to examine the smear for staining, there is no hard and fast rule say 10 drops or 6 drops or 7 drops, so as to optimally cover the smear. But you have to put it drop by drop, because excess smear will destroy this thing. So you have to put it drop by drop, and now wait for 2 minutes. After putting the smear, you have to wait for 2 minutes. This is called the fixation time. The blood, this 2 minutes is called the fixation time. It is the blood sample, this smear is getting fixed to that slide. Okay. Then we have to add either distilled water, not impure water, not tap water, either distilled water or something called buffered water. Now now we learn what is the purpose of counting the drops while putting the Leishman stain. Buffered water should be more or less equal to the amount of Lipscomb stain. That it is found that much is optimum for a good smear, which can be identify the cells under the microscope. So when we use the buffered water, why do we count the Leishman stain? We need not count, because there is no hard and fast rule about the number of drops of the Leishman stain, just to cover the smear optimally. But why we count the drops? Say we had required 5 drops, so we will also take 5 drops of buffered water. Say we had required 8 drops, we will also take 8 drops of buffered water. Okay. Here we are mainly, there is a supply of buffered water, but if there is no buffered water, also distilled water can be used. So what is how many drops of distilled water? If it is distilled water, then it is double the number of drops of Leishman stain. 5 drops of Leishman stain was required, so now we will give 1 like this, 10 drops of distilled water. Okay. Either the equal number of drops of buffered water or the double the number of drops of distilled water. These things have been standardized after experimentally. Okay. This is not on our own choice. This has been standardized experimentally. How the results are coming, then they have been decided, and then that is followed in all standard laboratories. So after we put Leishman stain, 5 drops, 6 drops, it may be 7 drops. If it is distilled water, 7 drops of Leishman stain, so it will be 14 drops of distilled water. 7 drops of Leishman stain, it will be 7 drops of buffered water. Okay. Now you have to know, you will be asked, plus you have to know what does Leishman stain contain. Okay. It contains eosin, methylene blue, nothing to write, everything is given in textbooks. If anything, I am not making things by myself. If anything, for the purpose of remembering, it is easier to remember something, you have to note down, I will tell that, but I am saying nothing from my own, I have not invented these things, nobody has. So it is in every standard textbook, it will be there. So what is, you follow any practical textbook, what are the components of Leishman stain? Eosin and methylene blue. These are the staining components of Leishman stain. Staining, those components which will stain different portions of the different components of the blood. So eosin and methylene blue. Okay. So eosin will stain what this comes, the name comes from eosin will stain eosinophils and RBCs. Particularly what part of eosinophils? Granules. Eosinophils contain granules, granules of eosinophils and RBCs. Okay, remember like this, eosin will stain granules of eosinophils and RBCs, and whatever else from the first day know this like this, whatever else will be stained by methylene blue. So in the exam, if you are asked, granules of neutrophils are stained by, and this is the era of MCQs. Sometimes granules of in Leishman stain, granules of neutrophils are stained by, so your answer will be methylene blue. So don't try to remember the components like this. So neutrophil, methylene blue, eosinophil, not like this. Just simply remember eosin is for eosinophils, granules of eosinophils and RBCs. And RBCs take a light, after the staining is complete, RBCs take a light pink color. Eosins, granules of eosinophils and RBCs are by eosin. This you may write down if you are willing, you may write down. Don't remember, try to memorize like this: neutrophil granules by methylene blue, basophil granules by methylene blue, lymphocytes by methylene blue, eosinophil by eosin. So there is so many things to remember, so many things in this vast course, that you have to remember. Whatever you can remember easily, choose the easier path. Eosinophil, granules of eosinophils and RBCs by eosin, and whatever else is by methylene blue. Okay, so these components of Leishman stain are staining the different components of blood. Granules of eosinophils and RBCs. RBCs are devoid of nucleus. RBCs will be it will be visible just a single rounded shape. RBCs by eosin. Which component? Eosin. And everything else, granules of neutrophils, lymphocytes, monocytes and no granules, total this cell will be stained by methylene blue. Okay. Now how to identify? For the beginners, you have to identify the RBCs and different types of WBCs, platelets at a later stage, not now. RBCs and WBCs. Now after you stain, what is the, we have just completed a part of the staining procedure. That means you put the stain, you count, just keep in your mind the number of drops, then you wait for 2 minutes, then you put the distilled water or buffered water, whatever is available. If buffered water, equal number of drops. If distilled water, hello, হ্যাঁ একটু পরে করছি, ক্লাসটা নিয়ে নিই, একটু পরে বললে হবে, ঠিক আছে। Distilled water, equal number, double the number of drops buffered water, and now keep for 10 minutes. This is the staining time. First 2 minutes was fixation time. The smear was getting fixed to the slide, and now is the staining time. Actual staining is occurring. Okay. Now after these 10 minutes are over, now see what I had told, you will require time during the classes, come on time, during learning, during the exam. Here just with these 2 minutes, you have to wait 1.5 to 2 minutes. For allowing the staining to occur properly, you have to wait for 10 minutes. These take 12 minutes. These itself take 12 minutes. Before that you have to take the apparatus. During the exam, total time allotted is 45 minutes. You have to collect your apparatus, prick your finger, and do whatever you are doing. Total time 45 minutes up to getting your result. So 12 minutes is just for the fixation and staining. So you will require time, and when you learn in the class, perform in the class, this will require time. That's why practical sessions are of 2 hours. Okay. So if you don't start on time, it will be difficult. So after these 10 minutes are over, you will find it will take the color. It will appear somewhat bluish green, greenish. So a greenish, there will be a scum. Scum means a layer will be formed. You can see it. So that you have to access, that you have to discard. Now the smear has been fixed, it has been stained, and excess fluid you have to discard. Better just gently use tap water. Don't you we have to use tap water. Don't allow tap water in full force to fall on the slide. It will wash away everything. What is the method? The end where the blood drop was taken, you just put your thumb over there. The thumb has a somewhat a greater surface area. This allow the tap water to fall very slowly, if possible in drops or very slowly over this. Every step is important. If your smear gets washed away, you can do nothing. You have to repeat the whole process, and your time will be over. At least for exam purposes, if you lose time, then you are losing 10 marks. Out of 100 marks, 10 marks are for the hematology experiment. So what is the precaution to be taken? Then you allow tap water very gently to fall on the thumb. This is recommended. Whatever your purpose is to wash away the excess fluid. Allow water to just gently fall over the thumb, and it will just trickle down and wash whatever is, you may say, extra, whatever is not required, not needed. But if the smear gets washed away and then you put the slide under the microscope, you watch nothing, then you have to do the whole procedure. And if you don't wash this away, then also it will create problems. So you have to wash this. It is called the greenish scum is formed. You have to wash away that, but with that you have to do that very gently, with carefully. And now after this has been done, we allow it to dry. We allow the film to dry. This is now a stained blood film. This is called now it will be called a stained blood film. We have to allow it to dry before looking at before watching it under the microscope. Now this drying will require some time. So see, if you don't start on time, then you will lose time in the exam, of course, and also in the class. You have to come on time and do these things. After drying, you will watch it under the microscope. So already say almost 25 to 30 minutes are over. Now you have to identify cells, count the leukocytes, and then prepare your result. Okay. So 45 to 50 minutes will be required. Allotted time is 45 minutes. So for this experiment, differential leukocyte count. Now what is differential leukocyte count? It is the number of different types of leukocytes expressed as percentage in a given blood sample. In a given blood sample, the number of different types of leukocytes expressed as a percentage of whole. Whole means the total number of leukocytes. So more the number we count, more accurate will be the figure, but usually we count 100 cells. Now for counting the cells, first you have to identify. Now what do you think most of these film which will be the most abundant cells, most number of cells? RBCs, of course, they are huge in number. Theoretically we know RBCs in the human body are say around 4.5 to 5.5 million per cubic millimeter of blood. Cubic millimeter, or also called microliter. Cubic millimeter or microliter. I don't think I have to teach mathematics here, but cubic millimeter and microliter is same. Microliter is SI system, standard international system, and this cubic millimeter, this is millimeter, centimeter, this is in the CGS system. So cubic millimeter and microliter is same thing. Blood cells, count of blood cells, total count of blood cells are expressed in cubic, in our country in cubic millimeter or microliter of blood. Like hemoglobin, accepted normal standards of hemoglobin in an adult male, say 14 to 16 gram per, that is in deciliter of blood. Cells in microliter of blood. It is standard expression is like this. So RBCs are 4.5 to 5.5 million, say around 5 million per microliter of blood, and WBCs 4,000 to 11,000 per microliter of blood. That means RBCs are almost more or less 1,000 times, 500 times, 1,000 times in number to WBCs. Platelets 1.5 to 4 lakh per microliter of blood. Okay, so in the blood film, when we are watching, the cells can be distinguished after staining. Different components has taken up different stains, either iodine or methylene blue, and are now distinguishable. The most abundant thing in this whole film will be RBCs, and they will appear as rounded pinkish figures, rounded, although actually RBCs are biconcave in shape. We will have theoretical discussions. RBCs are biconcave in shape, okay, with the central part is narrower, okay, the diameter of 7.5 micrometer. So these things, and in this era of MCQs, these informations are also important. But RBCs will be visible almost as rounded pinkish figures in the, and most abundant, whatever is most abundant in a good blood sphere, it is the most abundant cell. Obviously, these are more in number, and WBCs in every field. So what is ideal smear? What is an ideal smear? Ideal smear is, it will be, one point we have told, it will be tongue shaped, and one point we have told it will be 2/3 of the, more or less 2/3 of the slide on which the smear is prepared. It will cover the 2/3 of the length of the slide, and it will be tongue shaped. But that means from the end of the origin where the blood drop was taken towards the other end, it will gradually taper. Okay. Now, when we look under the microscope, first we look under the low power objective. So, what is the low power objective? Objective lenses. There is a lens in the eyepiece, and there is a lens in the objective, whichever objective we use. Low power objective means it will magnify it. Usually the low power objectives magnify the object 10 times. Why these are objective lenses? Because it is among the lens system of this microscope we are using, it is nearer to the object. So these are called objective lenses. Now they are of usually 3 or 4. 3 we use mainly. One is the low power objective, which will magnify the object 10 times. The one which magnifies is 40 times, is called 40 or 45. It is written over that is the high power objective, and another is called the oil immersion objective, because for using that we use oil. Okay, cedarwood oil. Cedarwood oil, why we use cedarwood oil? Oil immersion objective, for finer details, it has a very fine aperture. Okay, now if we don't use cedarwood oil, what is there between our object? Object means here it is the blood smear over the slide and the lens. There will be a gap, and what will that gap contain? That means it contains nothing. Nothing means air, but refractive index of air. This knowledge of optics is better. My knowledge is much worse than yours. Your knowledge is better. You have just read physics and come. So if the refractive index varies, then most of the light rays will get diffracted, but aperture is small. For finer detail, aperture is small. So what is used? Cedarwood oil between the slide and the lens. Lens means objective, lens of that objective. Why the cedarwood oil is used? Because more or less it has same refractive index as that of glass. Slide is made up of glass. So now it eliminates that layer of air. Now the more rays are, even if the aperture is small, more rays are coming. Rays are not diverging away. That is the purpose of using cedarwood oil. Okay. Any substance which has more or less similar refract that of glass, as the slide used is made up of glass. So glass slide, cedarwood oil, when you use the oil immersion objective, then it is best to view for counting leukocytes and getting the result of the differential leukocyte count. Best is to use oil immersion objective. High power objective can also be used, it also gives the result, but more details and everything will be visible properly if the oil immersion objective is used. But first use the low power objective, and whenever the magnification is less, you get a greater field. This is from common sense. Visiting somewhere and taking pictures. If we zoom in, then the field will be lesser with the help of your mobile camera. If we zoom in, the field will be lesser, the area which will be covered will be lesser. If we zoom out, then the field will be greater, a greater area will be covered. Same thing, when we are using the low power objective, more area of the blood film can be seen at a time. When using the high power objective, that is now it is 40 to 45 times, and now it will be somewhat lesser area. Using the oil immersion objective, the magnification in 100 times, still lesser. And what is the total magnification? This will be asked. What is the total magnification? Magnification of the eyepiece lens over which eye is put. Here there is a lens, eyepiece lens multiplied by the magnification of the objective lens. Now during the exam, out of all these things, this simple answer, sometimes it comes, it will be addition, not addition, it will be multiplying. That multiplies it 10 times. Again, those rays are getting refracted, we are again getting refracted through the eyepiece lens, and then it is again getting magnified. Whatever is there, first 40 times, say using the high power objective, 40 times, then again 10 times, then total magnification is 40 into 10 is 400, not 40 plus 10, 50, 40 into 10, 400. Or when using the oil immersion, then it is 100 into 10, 1000 times magnification than the actual object. Here we are counting the blood cells. So most abundant thing in the blood cells is RBCs. Now ideal smear, we talked about 2 points which we should aim at. That one is tongue shaped smear, should cover 2/3 of the slide, and in every high power field there will be at least one WBC. If we do not, we examine different fields, we move the slide. How to move, that was the first class on the compound microscope. How to move, this is the fixed stage of the microscope, and microscope holding the, whenever we hold the microscope, one support, we have to support it with one hand. That is the rule of holding the microscope. Now this is the fixed stage, this is the mechanical stage. Mechanical stage can be moved from one, either forwards or backwards, or from side to side. Now we are moving according to our convenience, we are seeing different fields, and we have to count the cells. So cells are counted from the origin of the, that means on that end where the blood was put, cells are counted, and then again we come like this. This board is not working properly. Like this I am showing with the finger. Like this we go towards that one end. Now what we will see at the edge, at the length of the, that means if the smear is tongue shaped, this is not very easy to understand. If the smear is shaped, we go from one end to this. Now as we move gradually further, what we will see, some areas will be blank, because if it is tongue shaped, some areas will be blank, because now we come down again, go from this end to this end. Like this we have to count several fields till we get at least 100 WBCs. Now ideal smear in a high power field, there should be at least one WBC. In low power field obviously there will be more, because it covers more larger area, but one high power field, one WBC, it's okay. In oil immersion you may not get WBC in every field, but RBCs will be there. RBCs are numerous. Okay, RBCs appear as pinkish, these pinkish rounded figures, those are numerous. In every field there will be several RBCs, so many RBCs, but WBCs will be, you may not get in every field, especially when we use the oil immersion objective, because more the magnification, less will be the area of the field. But still we go like this, come down, again go to the left, again come down, again go to the right, like this, covering the whole film. We have to count 100 WBCs, and then note the different types and how many among 100, how many are neutrophils, how many are lymphocytes, how many are eosinophils, how many are monocytes, how many are basophils. Now basophils are very rare, like RBCs are the most abundant. Among RBCs and WBCs, rarest is basophil. So don't identify, for beginners, for your exam purpose, we would recommend don't identify a cell as basophil unless absolutely sure. Now how to identify the cells, that is the next part of the discussion, how to identify the cells. RBCs, by those It has taken up eosin pinkish stain. Eosin covers what? Eosin stains what? RBCs and granules of eosinophils. Everything else by methylene blue. RBC has taken up eosin, most abundant pinkish, and note the size. Now in the exam, how do you identify this is RBC? So the size, if you answer the size is, so the size of the RBCs is 7.5 micrometer. Is that a correct answer? Correct or not? Yes, this is correct. The information is correct. But the next question from the next statement of the examiner will be, have you measured it with the scale under the microscope? No. So, whenever we are watching under the microscope, we are not measuring it. We are just assuming. Look at the size of the RBCs. So, first step to know is to look at the RBCs, and in your mind you get an idea that what is the size of the one normal, one singly, what is the size of the RBC. Now, while identifying the WBCs, first step is comparing the size. How do you identify it is a WBC? Firstly, it will be taking up different staining, and it will be, you can make it out from the color. But when you have to describe what WBC it is, you have to compare it with the size of the RBC. Sorry, you have to compare it with the size of the RBC. Not like that from your theoretical knowledge, the neutrophils are almost more or less 1.5 times to double the size of RBCs, and from your theoretical knowledge you know it may be 10 to 14 micrometer. Micro means 10 to the power minus 6. Okay. 10 to 14 micrometer. But during the exam, if you utter this, then next point is whether you have measured it under the microscope. It is not possible. So WBCs are different types of WBCs. First point to identify is comparing that with the size of the RBCs. Largest WBC is the monocyte. Largest WBC. It is almost 2.5 to 3 times of that of the RBC size. So first you get an idea RBC size. I have made a blood smear. On the first day it will not be a very proper ideal blood smear. It is not possible humanly. Now when you go on this next day when you make the smear, first day you make, next day it will improve. So in a blood smear, how the RBCs are looking, try to get an idea of the size. So largest is the WBC is monocyte. It is almost 2.5 to 3 times of RBC. That you are just looking at and getting and assuming, not you are measuring with the help of a scale. So don't do, in the practical portion you never utter that this is the size, this 14 micrometer, 20 micrometer. Yes, if it is 3 times, then RBC is roughly, the diameter is 7.5 micrometer, then monocyte diameter will be 20 to 23 micrometer. But you are not measuring it actually. Here we are looking at and assuming, comparing the size of the WBC with the size of the RBCs. RBCs are most abundant, so not difficult to compare. When you see a WBC in a field, surrounding it will be so many RBCs. So just you are looking at and getting an idea. There are other points, we are coming to that. But first, WBCs, types of WBCs are to be identified compared with the size of the RBCs. So neutrophils, also eosinophils, basophils are almost 1.5 times to 2 times somewhat larger than the RBCs. Small lymphocyte we spoke of, according to size it is small or large, can be classified. Small lymphocyte is almost the same. It is not exactly same, but almost the same size of RBCs. Large lymphocyte is almost twice the size of RBCs. Monocytes are still larger. It is more than twice, but it may be that one single monocyte is twice. So that is just an idea. You have to use other salient point, other points to identify how is the nucleus looking like. We are coming to that. But first, for the beginners, when you learn to identify like this. In the books when you will read, it is given 7 micrometer, 14 micrometer, but when you are actually working hands on, you can't measure. You have to just compare with the size of the RBCs and identify. So WBCs take up, mostly what will they take up? Which part of the stain? Methylene blue. methylene blue part of the stain. So neutrophils, how do they look like? First compare with the size of the RBC. Okay, this is the WBC, this may be this. Neutrophils, RBCs are non-nucleated but WBCs are nucleated. How will the nucleus of WBCs look like? This is different for the different types. Neutrophil will be a multiple lobe nucleus, usually 2 to 7. Still now we know this is 2 to 7. This may be the nucleus is lobe nucleus. Okay, 2 to 7. Eosinophil generally 2 lobes, not more than that. And after you go on seeing, when you go on looking at some eosinophil when you have found, you will see that, you will observe that it is somewhat spectacle shaped. Spectacle shaped bilobe nucleus, it appears such that looking like the spectacles put on the eyes of a human being. It is spectacle shaped nucleus. It is described like that. Eosinophils. There are other points to identify. Only one single point will not be enough, or you have to, at least for you when in the learning phase, nucleus, if you are seeing 3 or 4 lobes, it will be visible. If 3 or 4 lobes, then more it is going more towards neutrophil. 2 lobes, it can be either neutrophil or eosinophil, but eosinophil is spectacle shaped. And how to differentiate now? You have found 2 lobes, more or less same size, that is 1.5 to 2 times the shape of RBCs. But it has taken up methylene blue, it is not pinkish like the RBCs. Now the granules of eosinophils has taken up eosin, but the nucleus has taken up methylene blue, right? In eosinophils, granules will take up eosin, so pinkish coarse granules. Granules in eosinophils are coarse, but in neutrophil it will be finer granules. Understand? This is through theoretical discussion, just you are having a very preliminary idea. When you look at the cells, you can understand that this comes by practice. How to identify? You have to stand your own observation. If you say eosinophil in front of the examiner, examiner will ask why you are saying that. If you can stand your observation, you are correct. Okay. Obviously, a 7-lobe, 5-lobe nucleus, WBC with a containing a 5-lobe nucleus, it will be a neutrophil. Eosinophil does not have 5 lobes, so you cannot stand it as eosinophil. But if there are 2 lobes, you will look at the other points and then come to your conclusion that okay, this is then a neutrophil, this is a eosinophil, this is a basophil. Again I say, for all practical purposes, for the exam purpose, try not to utter a cell as a basophil, as a WBC as basophil. Our basophils are very rare. In standardized reports, investigation reports, you will see basophil is, in most cases it is written as 0%, because studying the smear, the technician found no basophil, could not find any. So basophils are very rare. It is theoretically accepted is 0 to 1%. So if not very confident, basophils will have some features. Granules are fine, lobes are 2 or 3, and it will be thus 1.5 to 2 times the size of the RBCs. But basophils are very, very rare. So unless very confident, don't utter it as basophil. Examiner may cross you and prove that you are incorrect. It should be a it was either a neutrophil or a eosinophil. 2 lobes, spectacular shape, coarse pinkish granules, eosinophil. Fine granules, okay, not so pinkish, between granules of neutrophils will not take up eosin. So that is all. Fine granules with 2, 3, 4, 5, 6, 7 lobe nucleus, it is neutrophil. Neutrophil, eosinophil, for all practical purposes, don't utter basophil unless very confident. Lymphocyte and monocyte. Lymphocyte is now in lymphocyte and monocyte where there are no granules, nucleus is also not lobed. This is one feature, structural feature. Nucleus is not lobed. Lymphocyte, rounded nucleus, almost covering the cell membrane. There is a cells will have cell membrane, and the nucleus is inside it. It is a rounded nucleus almost covering the whole of the cell inside. whole of the cell, so that there will be a thin rim of cytoplasm visible in the periphery. So it is two round, one round, and inside that one rounded shape is another rounded shape. Inside it, that is, it has taken a methylene blue. Okay, so you will identify it by the color also. Rounded, the cell membrane, inside that is the nucleus, and it has almost taken up almost the whole of the cell. But there are deviations. It is not like, again I am repeating, not like mathematics 2 plus 2 4. It will be half of the cell, 2/3 of the cell. Sometimes it may be smaller, but in most cases it will take up the whole of the cell. So the outside the cell, so inside one spherical shape, if another spherical shape taking up almost the whole, then there is a thin rim of cytoplasm in the periphery, and by this you will identify large lymphocyte will be, by looking at, by your experience, large lymphocyte will be more or less twice or somewhat greater the size of the RBCs, surrounding RBCs. There will be, again, when you are actually looking at, there will be many surrounding RBCs and maybe maximum 2 or 3 WBCs in one field you are looking at under the oil immersion microscope, under the high power field. So lymphocyte we identify like that. If the lymphocyte is a small lymphocyte, then it may be similar to the size of the RBCs. Okay, it is a small lymphocyte. But if a large lymphocyte, then it will be twice. But whether small or large, then you have to identify with it is a non-lobe, not a lobe nucleus, rounded nucleus almost covering the cell. And monocyte, how do you identify? First point is comparing with the size of the RBC, it is a large cell and comparatively larger cell, almost maybe 3 times, 2.5 to 3 times the size of the RBC, and here the nucleus is usually bean shaped or kidney shaped. This is one very prominent identifying feature for the monocyte, bean shaped or kidney shaped nucleus towards one side of the cell. So the rest of the cell is cytoplasm. In lymphocytes, it was a very thin rim of cytoplasm outside the rounded nucleus. Okay, that rounded nucleus is covering almost the whole of the cell. But in case of the monocyte, large lymphocyte or monocyte sometimes be similar in size. Now identification point is, in monocyte it will be a bean shaped or kidney shaped nucleus which is towards one side of the cell. It is peripheral in location, and the rest of the cell you can identify the cell membrane. One side there is the bean shaped cytoplasm or the kidney shaped nucleus, not cytoplasm, nucleus. Rest is the cytoplasm of the cell, and it is that cytoplasmic part. In lymphocyte, cytoplasm is very less, thin rim. It is difficult to identify any more point, but monocyte, if there is cytoplasm and nucleus is easily distinguishable, you can see the considerable part of the cytoplasm. Will it contain granules? No. But in case of the nucleus and neutrophil, eosinophil, basophil, it is better not to say, but neutrophil and eosinophil, it will contain granules. Granules of eosinophil will be pinkish and coarse, it has taken up eosin, and granules of neutrophil will be fine. Lymphocyte, monocyte, no granules. No question of any granules in the cytoplasmic part. So these are the points on the basis of which you will identify, but very common mistake, don't speak any figure about the size, 7 micrometers, 10 micrometers, 14 micrometers. It is not possible to say because we are not measuring it into the microscope. We are just comparing with the size of the RBCs. These are how to identify the WBCs, and now comes counting. How to count? You make a box of, you have to produce the result. Ultimately, you have to produce the result. So as soon you make a box, one method is you make a box of 10 into 10. That means one square box, 10 rows, 10 columns, and so 100 squares. Inside the large square there are 100 squares. You make it on a piece of paper. As soon as you find the first WBC, nobody knows when you are identifying the cells what will be the first WBC which you are seeing. Maybe it is a neutrophil, maybe it is a lymphocyte, maybe it is a eosinophil, anything. So in one box you write as N. If you find the first cell as neutrophil, one box you write as N. Next you find say some lymphocyte, you write an 1L. After you have completed the 100, so again when I am describing this in few seconds. You can understand that this drawing of the box and then seeing the cell, then writing it, all this required time, and total time is 45 minutes for your experiment. So you have to practice in the class, and during the exam you have to produce. You have to be fast and you have to produce. Any method, any step is wrong, then you will be, any step if it is wrong, then you will not be able to produce the result. Well, you have to repeat the staining, then time will run out. If you are repeat the staining proper, total time will run out. It is not possible to complete in 45 minutes. So differential leukocyte, now you write N, in another box L, then E, then again say one neutrophil comes N. At the end you have to count. At the end you have to count how many neutrophils, how many eosinophils. Basophil I am considering there will be no, there will be maybe one or maybe no basophil. Monocyte and lymphocytes. No need to differentiate, write a small lymphocyte, large lymphocyte separately, lymphocytes. Okay, at the end count out of 100, my result is neutrophil say 60. Now accepted, theoretically we should know, which will be, we will learn in the theory classes also. Neutrophils is almost 60 to 70% usually. So usually we get, say, we have got, after counting we get say 62 neutrophils. Lymphocytes are usually 20 to 40%. Eosinophils usually 1 to 5%, 2 to 3% are monocytes, and basophils are very rare. Okay, this is how to count and do. For counting purpose, there is another method. Usually most students are using this box method. This takes somewhat more time, drawing the boxes, separating it into 100 smaller boxes. Another method is called, it is called the tally bar method. That is what you do, on a piece of paper write N, E, N, L, E and M, and if you wish you can also write B. Again I am advising you don't utter any cell as basophil, you write B. Neutrophil, say you, as soon as you get one neutrophil, put one vertical mark, you have written N, L, E and M. Neutrophil, lymphocyte, eosinophil, as soon as you get neutrophil, put one vertical mark beside where you have written N, put one vertical mark. Next cell you say get another neutrophil, put another vertical mark. You get one eosinophil, beside E you put one vertical mark. Every fifth, same cell, every fifth you cut it out horizontally. Four vertical marks, you have given one stroke. This is somewhat less time consuming than this drawing of boxes and all. That means 1, 2, 3, 4 and you cut it out horizontally means 5 cells. 5 cells of that type. Do you get it? This is called the tally bar method. At the end, count. They say 11 pairs of, not pairs, 11 sets of these 5 neutrophils. So neutrophil is 55. Or 11 sets and 2 vertical marks. That means 55 plus 2 neutrophil. Counting is easy, and in this tally bar method is less consuming. You may do, ultimately your aim is to get the result, you may do anyway. That drawing of boxes is somewhat more time consuming. Total time is very short, 45 minutes. This is how we have to perform. That means first step is cleaning the finger, collection of blood sample, drawing the smear, then what is the step? Staining the smear. Staining comprises fixation time 2 minutes and staining time 10 minutes. Then washing of that greenish scum, which will be formed very gently by allowing tap water to fall over the finger. Then washing of that thing. Then what do you do? Then allow it to dry. Every step is important. Allow it to dry. Now watch under the microscope. First under the low power, identify the RBCs. Get an idea how in the first class when you are doing RBCs, how they look like. Then what you do? RBCs, how they look like. Then you compare, you have, for identifying WBCs, you have to compare it with the size of the RBCs. After you see over the low power, see under the high power. Counting is possible under high power, but better to count for finer details for using the oil immersion, using cedarwood oil. Okay, in the first class, this topic will go on for at least 1 or 2 weeks for practice. So, more than 1 week. So, you do it, read the topic. From Monday onwards, you have to do it, perform it, read, come and read whatever has been discussed today. Study the topic, how to do. First step is on Monday you do it by yourself. Okay, you have to do it by yourself and complete the whole thing. In the exam, time is 45 minutes. Obviously, on the first day you will require more time, there will be mistakes. So, total session is 2 hours. On Monday you do this. On Monday and next Thursday, at least this is the way you are doing it. Okay, and every step has to be followed, every step is important. If you don't dry the finger before taking the drop of, that is pricking and taking the drop of blood, then the thing will be diluted. Blood will get diluted with the spirit you are using. So, I sometimes advise that bring your own needles, use it on your own. Needles will be supplied. Those are usually more in supply. Here are the lancet needles with which you will prick the finger. Capillary blood, 1 or 2 experiments are there which is not possible with venous blood. Venous blood, how it is taken? This tourniquet is used. Usually we take venous blood is taken from antecubital vein in the antecubital fossa. There is a very prominent vein which is the antecubital fossa. Anti means here ANTE, front of the cubital fossa. This part of the hand is the antecubital fossa. What is for, you will learn in anatomy. This is the antecubital fossa. There is one vein, one artery, one nerve. One artery, one vein, one nerve here. Venous blood is taken. Tourniquet is applied over the arm, and then blood is drawn from the vein. That is not possible to do by oneself. Many experiments are possible with venous blood, but 1 or 2 experiments from the exam point of view in your syllabus, you will come to know about those later, which are not possible with venous blood. Okay, capillary blood. So, from the first day, capillary blood is must for those. So, from the first day, you learn how to prick capillary blood. If that experiment comes in the exam, obviously there is no question that venous blood, you can use venous blood. So, and if the examiner, during the exam, everything depends on the examiner, most things. If the examiner requires that you do it, it may be given in the question, perform this such and such experiment, differential leukocyte count with your own capillary blood. Then you have to do it, so you have to learn how to prick the finger and take the capillary blood. Venous blood will not be supplied. The examiner wants, then you have to work with capillary blood. So, first or second day in the hematology lab, first step is learning how to collect blood sample, preparing the smear, then doing the differential leukocyte count by identifying the different types of WBCs. Okay. So, Monday onwards you start at least 2 or 3 classes for this topic, 2 or 3 practical sessions, maybe Monday, Thursday and next Monday. Okay. Next coming Monday is 11th, you do this, study and come. Study this topic and come to the class. If you don't study, and I would recommend for these practical sessions, carry your practical books at least for the hematology section. Then during the at least in the initial classes, you will find the book helpful. Bring your own practical textbook for the at least for the hematology sessions. Okay. So, for today it is over. Remember, still now you have not been allotted roll numbers. You have to sign. You have to sign your names before leaving. That is your attendance. Some people have lost attendance in the last 3 days. Either they did not know. If you don't sign right during or after the class before leaving the session, your attendance is counted as 0. And this is very hard